All right, aloha guys. My name is Hanale Ho'opai-Silva. I'm with the Coral Resilience Lab, which is part of the Hawaii Institute of Marine Biology out in Kaneohe on the island of Oahu. And today I want to talk about a little project that we're working on titled Restore with Resilience, where we try to proactively restore the reefs using thermally tolerant corals. As we all know, coral reefs are being constantly threatened by anthropogenic natural stressors, which is why any efforts to restore the reef is important to keeping this habitat from going under for good. And we also know that ocean temperatures are slated to rise over time, which is why we at the Coral Resilience Lab wanted to determine that if we outplanted thermally tolerant corals on a reef, would that help improve the resiliency of our local waters and local reefs. So we're trying to determine this by planting corals that we deemed as having low and high thermal tolerance, and we're monitoring any changes or growth using structure from ocean photogrammetry. I'm going to get a little cultural. An olelo no eau, or Hawaiian proverb that's always stuck with a ocean-loving local boy like me, is he pu ko'a kani aina, which means a coral grows into an island. Hawaiians of old understood the importance that the reef plays in living a sustainable life on the islands. And Hawaiians of new and old practiced gathering food and resources from Mauka to Makai, from the mountain to the sea. And they understood long ago that without the health and without Mauka, there is no Makai, and vice versa. As a local fisherman myself, I understand how important the health of the reef is to keeping a reliable and necessary food source thriving, which is why working on a project like this is quite important to me, as well as it is to many other local people who rely on this natural food source and other natural resources that the islands provide. We're hoping to be proactive with this restoration, which will allow us to try and combat any extreme bleaching events that will most likely hit our reefs in the coming future. Coastal erosion, as well as sea level rise, is also a glaring problem throughout all the islands in Hawaii. If you take a look at this map, I should have made it bigger now that I look at it on the screen, but here's the potential economic loss that the island of Oahu may face with projected sea level rises. As we all know, coral reefs can dissipate a lot of wave energy. So outplanting corals as a means of restoration is a natural and organic way of increasing the size of a reef, which in turn can increase the protection that coasts get. So the corals that we outplanted are what we call corals of opportunity, which, if you don't know, are basically corals that aren't attached to any substrate on a reef and are free rolling. NOAA pre-identified these corals, and they invited us to test the resiliency component of this project. We did that by taking biopsies of the coral colonies that they gave us and taking it back to our lab for stress testing. In terms of stress testing, we did this by taking those biopsies and putting them in our saltwater tanks, where we introduced them to a steady increase of temperature from 24.4 degrees Celsius to 28 degrees Celsius over a span of 28 days. We took pictures of these fragments every other day to analyze and compare how they did with bleaching over time. And after 28 days of stress testing, we seen how our fragments did under heat over time. We gave them each a score depending on how they did, and because, as we all know, coral species bleach at different rates. Different coral species bleach at different rates. We made sure to compare corals by the species that they're in, and not as one just giant set of corals. So here's what a couple of our fragments look like. On the top, we have a rack full of biopsies, and on the bottom, we have that same rack, the same biopsies, but just after 28 days of heat stress testing. As you can see, a lot of the corals didn't fare too well against the heat, but we were able to see some corals that kept majority, if not all, of their pigment over time. I just wanted to throw this picture in here to show that even in the ocean, different coral species and even similar coral species will bleach at different rates. So it was cool to see that happen in this experiment within our lab. After analyzing all the pictures we took of bleached fragments, we determined which coral fragments held up against the heat and scored each biopsy on a scale of 0 to 3. 0 being lowest thermal tolerance and 3 being the highest thermal tolerance. With those scores, we were able to come up with figures like this. These two figures represent the distributions of the two coral species that we had the most of, Montipora capitata and Parietes lobata. as well as the 0 to 3 score that we have on our x-axis. As you can see, we have a fairly even distribution of scores. So to determine what corals had high and low thermal tolerance, we just used what side of the mean they're on. Left side of the mean corals we deemed as having low thermal tolerance. Right side of the mean corals we deemed as having high thermal tolerance. We wanted to split these up because we want to compare them once they hit the reef and see how they did against each other in case of a bleaching event. The corals that we outplanted were collected on a fringing reef right outside the Honolulu airport, so that's where we also planted them. 425 corals in total were outplanted, and we split them into plots respective of how their test scores were given. So 7 plots had the high thermal tolerant corals, 7 plots had the low thermal tolerant corals, and we photographed 8 control plots that had no outplanting done on them to try and compare how corals that are already on that reef fared up against our outplants. We have a total of 7 time points now, and we took photos every 4 to 5 months, and we use these photos to compare and analyze how our outplants did over time. With those photos, we processed them using AgiSoft Metashape, and we're able to come up with georeference orthomosaics as well as digital elevation models. You guys look here at the top right, here's a picture of what an orthomosaic, one of our plots, looks like. And if you guys look closer, you can see the red shapes. Those are the outlines that we did of our outplants. After we process all our photos and turn them into orthomosaics, we're able to manually locate and outline each of our outplants, which would give us metrics to collect, such as the survivorship of them, surface area, height range, as well as the volume. So the first thing I want to look at is the survivorship. Here in this figure, we have an x-axis that represents all the coral species we outplanted, separated if they had high or low thermal tolerance. And our y-axis represents the percent survivorship over the 20 months that we've been testing this. And as you guys can see, majority of our coral species show 100% survivorship, except for Parietes lobata and Parietes compressa, which still showed quite high survivorship. As I talked on before, we had the largest sample size among Tipora capitata and Parietes lobata. So moving forward and talking about our metrics, we're just going to be highlighting these 2 species. First metric I want to talk about is the surface area. Again, we have a very similar figure with the x-axis representing the 2 coral species that we're looking at, separated by high and low thermal tolerance. And then our y-axis is the percent change in surface area. So, for example, if we saw 100% change, that'd be like if our coral grew twice its size from its original outplanting. Surface area in this instance, we're counting as surface and habitat that surface and the habitat space that the corals are providing. We can see a significant difference in growth from the higher thermally tolerant capitata compared to the lower thermally tolerant, but we don't see much of a difference between high and low thermal tolerance in Parietes lobata. Spoiler alert, that's kind of the pattern that stays within these metrics that we're going to be looking at. So yeah, moving on to the area, which is the coral coverage across the reef floor. So just imagine you're looking down at a top-down angle of a coral and want to see how much space it's taken up. We can see a significant difference in the higher thermally tolerant capitata compared to the lower thermally tolerant capitata, but not much difference is found between the 2 categories for lobata. Moving on to the height range, which we counted as any upward growth that the coral underwent. It's not significant, but we do see a slight increase in growth from the higher thermally tolerant capitata. And again, Parietes lobata doesn't show much difference at all. And moving on to volume, which is the space filling growth that the coral has, we don't see much difference at all between the high and low thermally tolerant capitata. And it's not significant, but there is a slight increase from the lower thermally tolerant Parietes lobata versus the higher thermally Parietes lobata, which surprised us. But it's important to point out that whether they had high or low thermal tolerance, all the coral species that we tested and throughout all the metrics that we looked at, there was still positive growth overall, which is good. It's exactly what we hoped for in this project. And it's also important to point out that the reason of testing this was waiting for a bleaching event. Luckily, we haven't had one since we tried this experiment. But when a bleaching event eventually does hit Hawaii, then that's what we're going to be looking at and hoping to zoom in on looking at the high and low thermal tolerance of these corals. So yeah, in the end, we just want to make sure that the local reefs in Hawaii are prepared. And that's why the outplanting of these corals, we're just hoping to encourage reefs to be more resilient and self-recover. again, our goal is that if we can outplant thermally tolerant colonies on a reef, then eventually those colonies can give spawn to other thermally tolerant corals, and that will allow the reef to self-recover without much of our help. I kind of want to stress the importance of using photogrammetry on a project like this. Structure for motion photogrammetry allowed us to accurately measure and monitor all our outplants. It lets us keep a large database of past time points that we could always go back and check if we need to. And it just saved us a lot of time and a lot of money from having to go out and manually measure these corals ourselves. Overall, large-scale coral restoration is fairly new in Hawaii. We're definitely working out all the kinks in our project. We have a lot of improvement to make, and we're continuing to improve our methods to eventually implement in our future, or in our project moving forward. Looking ahead, we still have a lot of data that the structure for motion photogrammetry can give us. We've yet to look at the effect that depth plays on our outplants, if there is any, as well as any bleaching effects that may have happened over time. And I'm happy to say that we are expanding this project to different reefs on Oahu as well as different islands in the chain of Hawaii. And that will allow us to look at key differences between reefs and the reefs at different islands in Hawaii. Can let us look at if there are any key or specific environments, habitats that are that allow us to efficiently outplant, or as well as if there's any coral species that would work best for a project like this. And yeah, my PI warned me that I'd probably go through all my slides fast, and I'm well under time, so I apologize about that. But I want to thank you guys all for being here. I want to thank everyone at the Coral Resilience Lab. Thank you to all our partners, such as NOAA, DAR, Malama Mauna Lua, and Kuleana Corals. And thank you to our funders as well, National Fish and Wildlife Foundation. Thank you guys again. I'll take any questions if you have them.
We definitely have time for a couple of questions if anyone has anything they wanted to ask.
Yeah, great talk. And first off, I was thinking growth and thermal tolerance might not always be related. They might be decoupled.
Mm-hmm.
So you took these tolerant and less tolerant corals and you just looked at growth. Did you compare to the control groups to see if like maybe there's just no difference because we're not looking at a tolerance perspective yet, just overall growth?
Yeah, so that's a great question. We haven't looked at control yet. We're still trying to figure out what the best way to go about that is. We don't know if we want to test the control corals and see if they're thermal resilient or not, if we just want to test them straight out. So great question. We're still trying to figure that out. Hi.
This may have been missing your methods, but I just want to make sure. So you took corals of opportunity, right?
Mm-hmm.
And then you waited for a period of time to see the ones that survived, and then you outplanted those. Is that true?
Yeah, so we took the biopsies of those corals, stress tested biopsies, which we determined that would let us determine like the resilience of its parent colony.
Okay, but then everybody that you had, you outplanted from that?
Yes.
Whether they were lower or higher?
Whether they were lower or higher, yeah.
Never mind, then I don't have a question. Thank you.
Yeah.
Yeah, it's a really great talk. I'm curious if do you think any of the corals of opportunity are related or clones? And do you think that would be something worth looking at if you have samples?
Yeah, I can't for sure say, but I would definitely expect them to. They came from the same reef, and there wasn't a big area of, I guess, collection that happened. So I would assume that most, or not most, but some of them are definitely related, yeah.
Can I just ask a quick question as well? Yeah. On the point of corals of opportunity, obviously you've got choices of taking opportunity corals or sampling from parent colonies established on the reef. Do you think that just sampling corals of opportunity might sort of almost bias the starting fitness of the overall potential that's out there at all? Or?
I'm sorry, could you repeat that question?
So I guess where I'm going at, I'm thinking from our perspective on the Great Barrier Reef, we've got often sample corals and fragments of opportunity for permitting reasons. We can't sample many donor corals. And you know, we have to be very careful with those opportunity corals, which ones are actually in the best state to then propagate. So I kind of wonder if you're in that situation at all, or do you kind of sift through the opportunity corals to work out if there are ones that are not suitable at all? Yeah,
so there's definitely corals of opportunity that once we collected them and tried to take biopsies, they kind of already died. We lost a lot of corals in that way. I know that in terms of NOAA, they didn't want to just take fragments off the reef because breaking coral reefs doesn't go well with locals in the islands. So yeah, I just know corals opportunity was the route to go in terms of starting a project like this, which is fairly new. It just seemed like the easiest to do.
Yeah, great job.
Yeah.
Great talk. Thank you.
Thank you.